Hello, good morning everyone. So the topic for our lecture today is overview methods in genomics and proteomics, which will cover both CM Bio 021 and also CM Bio 022. My name is Dr. Farisan Ahmad from Department of Neurosciences, PPSP USM. So in this topic, we will learn about the methods or the approaches that we have in genomics and also proteomics analysis in cellular and molecular biology experiments. So for the introduction, I think we should first remind ourselves about the genetics constituents, about the genome, and also the complete set of the DNA. Okay, another thing which is also important for this topic will be the understanding of the central dogma of life, which consists of introduction of the DNA, is transcription into RNA, and also translation into protein, which are heavily involved in cellular and molecular biology activities. So before starting any experiments, the most important things that we need to have is, of course, the genomic or the proteomic materials. So for having these materials, there are many sources available. You can have your experiments done on tissue culture, cells culture, which is in vitro studies. You can also have your sources of DNA, RNA, or protein from blood, from in vivo or animal studies, and also many more. After having your DNA, RNA, or protein sources available, then you need to figure out which approach you are actually interested to carry out the experiment with. You may focus your experiments working on the DNA, RNA, or protein by selecting the appropriate methods for your study. We will discuss further about these methods later in the following chapter.